Hello everyone and welcome to today's live broadcast, Inference of Gene Signatures Associated with Response and Resistance to Targeted Immunotherapies, presented by Dr. Shirley Liu, Professor of Statistics, Biostatistics and Computational Biology at Dana-Farber Cancer Institute in Harvard University. I'm Susie Valdez of LabRoots and I'll be your moderator for today's events. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type that question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, notice that you will be viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower right. If you have any trouble hearing or seeing the presentation, click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. Good news, this educational presentation is offering continuing educational credits. Please click on the button at the bottom left-hand corner and follow the process to obtain your credits. Please join me now in welcoming Dr. Shirley Liu. I will now turn the presentation over to her. Welcome, Dr. Liu.
Thank you, Susie, for that introduction. It's my great pleasure to introduce our work. And we are a computational biology group, and so we develop statistical models to understand cancer therapy. Nowadays, precision cancer medicine is really on the rise. People are very excited to use genomic approaches to better prescribe or diagnose or prescribe drugs for cancer patients. For example, at Dana-Farber, when patients coming in, once they are diagnosed, their tumors get a mutation profile with the hope that with the specific mutations identified on the tumors, we could find a better drug to the patient. And indeed, a lot of the patients either will get a specific targeted therapy or they will be put on a trial that oncologists are testing new therapies. Unfortunately, even with the tremendous cost that is associated with drug discovery, for example, on average, it takes 12 years and $300 million to develop a drug, and 2% of those development efforts will finally end up in the FDA-approved drugs. For a lot of cancer therapies, only limited response rates have been observed, and also patients only observe very limited survival benefits. For example, many targeted therapies, you observe just a survival benefits of 6 months or 12 months, and eventually a patient will develop therapy resistance. And some patients will not even respond to the therapy initially. And so we are hoping that, with the high throughput data available in the cancer community, we can develop computational methods that can help us predict which patients will respond to a therapy and which patients will not, and also whether eventually they will develop resistance to the therapy. The ideal data for this are mutation or expression profiles of patients directly from those clinical trials. For example, before the patient is treated with the therapy, we take the tumor, we do some molecular profiling. After the patient is treated with the therapy, if the tumor is there, whether it's shrinking or continuing to grow, if we can get a biopsy sample of those tumors, and especially later on, if the patient develops resistance to the same drug, we can get either surgical or biopsy samples from the patient and do molecular profiling of those, and then comparing the pre-treatment, post-treatment where patient is sensitive, and also post-treatment when patient become resistant, and look at their differences. That would be really the ideal data. Unfortunately, these kind of data are very, very difficult. Patients do not want to get surgical or samples, or ideally not even biopsy samples after their disease have progressed and the tumor metastases are resistant to the drug. And so we are wondering what other model systems are possible if we don't have patient data directly. For example, we can derive cell lines that are from the patient tumors initially, so we can grow these cells in the Petri dish, and then we can treat the cell. We know, for example, the cancer cells have certain mutations. We can treat the cell with a drug, and initially maybe the cell lines are sensitive to the drug, but after a period, say a few weeks or a few months, the cell will become resistant to the drug. And so we could do gene expression profiling of those cell lines that initially respond but later becoming resistant, and see what are the differences between the two. These are helpful data, although it's still very, very limited, what is available in the public domain. And so what is end up being very helpful is these kind of compound screen data that is, in terms of the number, increasing very, very quickly. So there are 3 published studies now where the data is available, and such efforts are increasing. For example, in the CTRP project, scientists they collected 824 different cancer cell lines and treated the cell lines with 500 different compounds. For the DGSC project, it's also kind of close to 1,000 cell lines treated with 250 compounds. In the CCLE project, it was like close to 500 cell lines treated with 24 compounds. And so these kind of data are still growing. Each cancer cell line has been carefully molecular profiled, so we know their mutation status, we know their gene expression status, and then when the cell lines are treated with each compound, we can measure when the compound are at a different concentration or doses, we can measure how much the cells are being killed by the compound, which is the sensitivity of the cell to a particular compound. So since these data are really growing, we actually have a much better basis to start looking at cancer drug response. One type of data that's actually coming out more recently, although right now it's still limited, is very, very promising, is functional genomic screens. So these include shRNA or siRNA screens, or CRISPR screens, or ORF screens. So the idea is here, we can grow also the cell in a huge population of, say, 100 million cells, and then using a lentivirus or overexpression vector to introduce things that go to the different cells, either overexpress, knock down, or knock out a particular gene, and then treat the cell with a particular drug, and compare with the cells not treated with the drug. We can ask when a particular cell is overexpressing a gene or knock out of a particular gene, would that change the cell sensitivity to the drug that's treating the cells? And so this is also very, very promising, although this kind of data right now is still on the rise. We're hoping that the more data will become available. And so for the motivation of the work on targeted therapy, we want to develop computational and statistical methods for data integration to be able to predict response to cancer therapy, or model cancer therapy resistance, and also understand the mechanism of resistance, and hopefully to identify combination therapies in the long run to overcome drug resistance. So as I mentioned before, for our particular computational model, we are looking at compound screens. As I mentioned, it's hundreds of cancer cell lines treated with like hundreds of different drugs. And so this is one example. For example, in here, every dot represents a particular cell line, and inhibition on the y-axis really measures how much the cell is sensitive to a particular drug. And in this case, we are using a BRAF inhibitor, or the data is actually BRAF inhibitor treating cells. And so in general, cancer cell lines that harbor a BRAF mutation, the V600E mutation, will overall be more sensitive to the drug. So as you can see, all the red dots which harbor the BRAF mutation have less sensitivity to the drug than has more sensitivity or more cell growth inhibition than those wild-type cells. However, if we look at the two genes, like in the left side, you can see with the EGFR gene, now we discretize the data into 3 groups based on the level of EGFR gene expression. And so you can see, even though the red dots in general are these BRAF mutated cells are more sensitive to the BRAF inhibitor, the sensitivity actually is influenced by the level of EGFR gene expression. Based on this left figure, you can see the higher the EGFR gene expression, the less sensitive is the cell to the BRAF inhibitor. On the right side is the opposite example where we are looking at the LEF1 gene, and you can see here the higher the expression of the LEF1, the more sensitive is the cell to the BRAF inhibitor. And so we wonder whether we can find similar effect in the genome by checking every gene using these compound screens. And so we developed this computational model. We call it computational analysis of resistance, or CARE. This is a regression-based method. So in this case, Y is the drug response or inhibition, whether the cell is sensitive to the drug. So a high number would mean the cell is very sensitive to the drug, and the cancer cells are dying after the drug treatment, whereas a low number would mean that the cell is not so sensitive to the drug. And then in this particular case, we need to only look at the drugs where the target gene is known, where we are mostly looking at targeted therapy. And so the status of the target gene is this X in the regression model here. So X most of the time is a mutation status. For example, using EGFR drug inhibitor to target cancer cells with EGFR mutation, BRAF inhibitor to target BRAF mutated cells. We could also use expression as kind of a quantitative model in here. And so among all the cancer compound screens, we need to select those specific screens where we do see, as the previous slide show, in this case, the BRAF mutation needs to at least separate patient response, the cell line response to the particular drug. And if we don't even see this, we can't use this model. And so on those cell lines where on the drugs where we do see the drug target gene status is separating the sensitive versus the not so sensitive cell lines, then we can look at the CARE model. And so here we are hoping to identify another, or at least evaluate every other gene P in this regression model. So P is another interacting gene. By itself, it has a coefficient B in here, which says, maybe another gene always have some effect on the drug sensitivity, but in addition, we care about this last term in the regression model, the CXP. C is the coefficient, X is the mutated or the target gene status, and P is the interacting gene. And so the idea is, if we look at the second line, let me use the pointer here. If we look at this part, you can see what we care about is on this regression, we're asking, let's evaluate a particular gene. This gene might have its own effect, like this base effect, but also maybe it is also interacting with the target gene here, C, which eventually will influence the overall drug sensitivity of the cell to the drug. And so on this figure on the right, you can see we care about the B itself, and also we care about the C effect. So usually if the C is less than 0, it means that when the expression of the gene is high, the sensitivity of the cell to the drug will decrease, whereas if the C is larger than 0, it means that when it expresses higher, the cell will be even more sensitive to the drug. And so, okay, sorry, let me go back. Oh yeah, so I can use this. Okay, so let's go back to the original example there. So remember, there are two genes we're showing before, EGFR and LEF1. And so in this case, you can see the, so for EGFR, the CARE score, so the CARE score is basically a normalized B plus C score on the coefficient in the previous page. And so here, we look at the CARE score. You can see here, on the EGFR gene is a negative value, which means when EGFR gene expression go up, the sensitivity to the cell to this drug actually decrease. Whereas with LAF1, it's a positive score in here, which means that when LAF1 gene expression go up, the cell will be even more sensitive to the drug. And you can see from the p-values, these are very, very significant. And so you wonder, you know, this is just two examples. You can always pick and choose. The fact is we don't actually pick and choose. We test every gene in the genome as the P with the target gene X, you know, whether it's a BRAF inhibitor for BRAF or EGFR inhibitor for EGFR mutation status. So once we know the drug, we know the target gene, we test every gene in the genome for this potential interaction. And so with the drug screen, we'll get a CARE score, which is a genome-wide score. Every gene gets a CARE score, which is what we show on this previous slide on the blue and red square. And so every gene has a score, and later on, if somebody has a cell line or a tumor, based on the expression of genome-wide gene expression, we can correlate this genome-wide signature with the CARE score, which is also a genome-wide signature. And if we see good correlation, we can predict that this patient actually will respond to the drug. Whereas if the tumor or cell line doesn't have good correlation, we will predict that it won't respond to us for the drug we're interested in. So in this case, rather than cherry picking specific genes, we evaluate a score for every gene in the genome and use it to predict drug sensitivity of that particular tumor to the drug. And so the effect of each gene, even if it's noisy from the compound screens, will not have overall dramatic effect. We take effect, you know, the overall correlation of the samples. And so as a gold standard to see how well this thing works, we look at cells where initially they were a sensitive cell line, and over time, when it's treated with the drug, the cell become resistant, and we compare the gene expression differences between the resistant line and the sensitive line. Look at those differential genes, and we use those genes as our gold standard to see among the CARE scores that are really high or low. We can also rank the genes by the CARE score, and we see whether we can recover those genes that are differentially expressed between the sensitive and resistant line. And so, you know, so we use this to evaluate comparing with, you know, just the correlation of the gene expression with the drug effect or with ANOVA. And you can see overall, actually, the CARE model works really well. This is evaluated on 3 targeted therapy for like BRAF inhibitor, HER2 inhibition, and EGFR inhibitors. And so this is still testing on resistant and sensitive cell lines. We want to see how well it actually works in patient tumors. So far, when people try to predict sensitivity to cell lines in these compound screens, the evaluation is usually just made on the cell lines. We want to have a higher standard to see whether our signature can predict the tumor response. So by looking at published clinical trials, for example, in this trial, there are 12 patients treated with the BRAF so the patient tumors have the BRAF mutation. We know that they already have the mutation. Then we want to see whether the patient will really respond to the BRAF inhibitor using the tumor gene expression. You can see using CARE on the left side, we can actually predict a pretty good response and also predict how long the patient will have a progression-free survival. On the right side, we compare a number of other approaches. You know, CARE has the best performance, and also compared to CRISPR screens, you know, we actually do a better performance compared to CRISPR-A, CRISPR knockout, ORF screens, shRNA screens, also chronically treated cell lines. And so actually the cell lines in here, in terms of patient survival prediction, it doesn't do as well. So the first one was on BRAF inhibitor. The next one, we look at another clinical trial where patients were treated with a Herceptin, which is or a lapatinib, which is a HER2 inhibition for patients in HER2 positive breast cancer. So here we don't have the overall patient survival. We only know whether the patient responded to the drug to have observable tumor shrinkage. And so based on this, we look at whether the care can actually predict whether these patients can respond. And you can see we have a good separation of those responders versus non-responders. And again, our method outperformed, the chronic treatment. Actually here, chronic treatment, you get a pretty good result. siRNA and ORF screens. In the next slide, we look at patients with the EGFR mutation treated with EGFR inhibitor, and again, using the CARE model, we can make a pretty good prediction and also do much better than the other even experimental approaches. And so you wonder, maybe these clinical trials have too few patients, which is true, I think, in most of the published clinical trials from academic settings where expression data is available. And so we now test the chemotherapy. So chemotherapy, although is not really like a targeted therapy, some of the chemotherapies that are tested on the current compound screens do have a known target gene. For example, paclitaxel is a very widely used chemotherapy, and existing studies have reported that the tubulin gene is really the main target. And we did look at the tubulin gene expression, and indeed, the patients with higher tubulin are more sensitive to paclitaxel, which tells us that we could use the CARE model on the existing chemotherapy trials of paclitaxel. And so here we collected 3 different clinical trials. Remember, so here the CARE model was trained on cell lines treated with the chemotherapy. And then based on the signature, we tested 3 clinical trials of patients. And tumors have also gone through gene expression profiling. And based on the expression and our CARE prediction from the cell lines, we predict whether the patient will respond or not respond to the chemotherapy. And you can see, indeed, we can have a good prediction on the left side that the responders versus non-responders show a good separation, and also the method seemed to work very much better than experimental approaches. So in here, the clinical data is patients that are treated in one of these clinical settings. Based on the signature, we want to see whether it can predict in the other. And so in here, you can see based on AUC, it does better, but based on accuracy, it doesn't actually outperform. And so overall, it seems like with the CARE model, even though it's derived from the cell lines, we have good performance even on the patient tumor trials. And so question is, these seem to be all known targets, with well-established trials. Can we predict something new? And so we tried that. So here we look at lapatinib for a HER2 positive breast cancer. It turns out that this particular compound has been tested in all 3 CCLE, CTRP, and CGP, 3 different compound cohort, and they have actually very consistent result, showing 2 genes, PRKD3 and AKT2, as having very negative CARE scores, meaning that if we can inhibit PRKD3 and AKT3, the cancer cells will be even more sensitive to lapatinib. We also look at the gene expression. So these were chronically derived cell lines where the cell line were treated for long term, and then look at the resistant cell lines differential expression, and you can see also here these 2 genes have a much higher gene expression compared to the sensitive cell line. And then the last column, we are looking at a actual clinical cohort, which is we look at the association of this gene expression with the pathological complete response in the lapatinib treatment cohort, and you can see they are negatively correlated, which also indicate that if the expression of these two genes are high, the patient will not respond so well to lapatinib treatment. And so we ask, so actually there are more genes available on this list. We look at these two because they have inhibitors available. Indeed, for AKT3, it has already been reported in another study. And so we now test PRKD3, which has an inhibitor, but no clinical testing yet. So we first want to do a genetic test to make sure that, we can, if we knock down PRKD3 using RNAi, indeed, on the left side, we can see we can knock down the PRKD3, and also on the SKBR3 cell lines with the Herceptin or the HER2 amplification, the cells indeed will be more sensitive to lapatinib. So genetically, we can validate this. Now we test the inhibitors. So there are two inhibitors we found that inhibit PRKD3. We want to see whether this drug actually works synergistically with lapatinib, because sometimes when you have the cell you treat it with a drug, it will be a little sicker. You treat it with another drug, it will be a little sicker, but you add them together, the cell will be a lot sicker. And so we want to make sure that the synergy is the co-treatment inhibition subtracted by the additive effect of the inhibition. We actually can see more than what we expect just by adding the two drugs if they don't synergize. And so so on the figure here on the right side, you can see if it's red, it means that this synergy score is positive, meaning that indeed lapatinib and the PRKD inhibitors show synergy. And so we try a different lapatinib dose and also try at a different PRKD3 dose. And you can see in general, we see a good synergy. And on the left side, you can also see the inhibition of the cell growth after we do the co-treatment. We later on also derived a resistant cell line in the lab ourselves. So initially, the cell line, the SKBR3 cell lines are sensitive to the lapatinib drug, but after prolonged drug treatment, the cell become resistant. And so with this resistant line, initially we do see that it's not sensitive to the drug, but then if we treat now together the SKB3 inhibitor as well, the PRKD3 inhibitor as well as the lapatinib onto this resistant cell, you can see the cell become much more sensitive to the drug combination and also showing a good synergy. So in terms of HER2 inhibition, in addition to lapatinib, which is a small molecule inhibitor, there is also trastuzumab, which is an antibody drug targeting HER2. And so in addition to test the combination of PRKD3 inhibitor with lapatinib, we also tested this combination with the antibody trastuzumab, and you can also see good sensitivity or good synergy of the drug, the two different drugs to the trastuzumab. So in summary, we found cell line compound screens that are done on a big cancer cell line cohort can contain very informative biomarkers that can predict clinical tumor response to either targeted or chemotherapy. And so we developed this CARE model, which uses an interaction regression to identify the effect of another gene interacting with the specific drug target to influence drug response and use it to predict clinical tumor response. From this analysis, we also identified PRKD3 inhibition as a way to synergize with HER2 inhibition, either by lapatinib or trastuzumab in the HER2 positive breast cancers. So this is the first part of my talk, which is about targeted therapy. The next part, I want to talk about immunotherapy. As we know, recent years, we see a lot of progress in immune checkpoint blocker development. So, in general, the idea is, patient tumors usually are recognized by the immune cells or the T cells, for example, and but then the tumor cells will express this PD-1 or the tumor will express PD-L1 or B7. These are ligands which actually interact with the PD-1 and CTLA4 receptors on the immune cells, like T cells. And so the T cells, even though it's recognizing the tumor as foreign and trying to kill it, the PD-1 and CTLA4, once it's interacting with the ligand, then the T cell become dysfunctional. And so the development of the immune checkpoint blocker is we develop, companies develop antibodies to block this ligand receptor interaction, which allow the T cells to get activated to recognize the tumor cells as foreign and attack it. And so the good news is that the patient who respond to these tumors, usually, first of all, these tumors have a lot of mutation. These are the hardest to treat tumors. But those patients who benefits, usually we can see prolonged survival. So patient can really be cured 5 years, 10 years, disease-free. The bad news is that only a minority of those patients respond. And this is an expensive drug, and there are some known side effects like autoimmune side effects. And so the question is really, can we predict before the patient were treated with a drug, whether they will respond, whether they will benefit from immune checkpoint blocker? And so far, many things have been reported. For example, whether the T cells are infiltrating the tumor. The second is whether the tumor has a lot of mutations to really allow the T cell to recognize it as foreign. The level of PD-L1, which is the level of this ligand on the tumor cells. And also recently there are some reports that the T cell receptor repertoire diversity might also influence patient survival. And so each of these studies are done on a small, pretty small cohort. And of course, the billion-dollar question is really, how do we really predict? It's even with existing biomarkers, there are still clinical trials that failed because there's no good biomarker to predict which patient will respond. And so it may not really show enough benefits in the clinical trials. And so we want to build a biomarker for immunotherapy response. The relevant data is large clinical studies with immunotherapy treatment. Unfortunately, so far the published cohorts have very limited number of patients and also limited number of cancer types. So far, immuno checkpoint blockers have only been used in very few cancer types. What we do have that, the data in abundance is large clinical studies without immunotherapy. We have these large cohorts. For example, in the US, there is the TCGA profile where we have profiled over 10,000, or scientists have profiled over 10,000 tumors, over 33 different cancer types. ICGC is an international effort, from Europe, Asia, including the US, and Target is kind of a pediatric tumor consortium for the US. But the thing is, none of these patients, when their tumors are profiled, these are surgically removed tumors. These are mostly primary tumors that are treatment naive. And so the patient hasn't been treated with anything yet. The question is, can we actually see something from those tumors to tell us what happens to the immune system? So our hypothesis is that the tumor immunes, even the treatment naive samples, maybe there are already some interesting information about the patient immune system or the tumor immune microenvironment, which can tell us about immunotherapy response. So far with immune checkpoint blockers, there are two hypotheses where it will not work. The first type is on the right side. These tumors, if you look at them under the slides on the tissue staining, the tumor staining, you can see that the T cells are not getting into the tumor. And so it's not infiltrating the tumor to recognize the tumor as kind of foreign with the antigens. And so these are called immune cold tumors. On the left side, so if the T cell can't get in, then it can't kill the tumor cells. On the left side is inside the tumor, you can see the T cells infiltrating the tumor but it's exhausted. Maybe it's trying too hard and eventually it's just too tired. And so these are called T cell dysfunction or T cell exhaustion. And so these are hot tumors or inflamed tumors, but the T cell is there, but it's not doing its job. And so we want to see, maybe the immunotherapy failed to work because either it can't get in the tumor or it gets in the tumor, but it's not doing its job because it's too exhausted or dysfunctional. And so let's first model things on the left side for the dysfunction. And so for here, we again now look at the existing TCGA data or the large tumor cohort data where hundreds of patients were profiled for these cancer types. For example, we see in the TCGA for melanoma and the triple-negative breast cancer, we look at the patient survival. The y-axis is the survival. And so this is survival curve. We can see the T cell, the cytolytic activity of the T cells. So basically, when the T cell level is high and also the T cells are activated, overall the patient have better survival. So these are the red lines, meaning that when their T cells are high and it's actually doing its job, it's activated, we do see a survival benefit. However, there is a condition. For example, we look at the CBL-B gene, CBL-B gene. This is a well-known T cell dysfunctional gene. And so you can see, this is again in melanoma samples. When the CBL-B gene expression level is very high, CD8 T cell doesn't even have any survival benefits. On the right side, you can see only when the CBL-B gene expression is low, we can see the survival benefit. When the CD8 T cell or cytolytic activity is high, the patient do have better survival. So by the way, here, the cytolytic activity, we use the gene expression level of CD8A, CD8B, granzyme A, granzyme B, and porphyrin level. So you can see this actually is similar to the previous care model. We derived this new model here. It's also regression. On the response here on the Y, here the H is the kind of the survival, the hazard of the survival curve. And the T here is really the level of cytolytic T cell activity, which is measured by these 5 genes expression level. And then we ask whether there's another gene, like CBL-B, which interact with the level of T cells or cytolytic activity T cells to influence the overall patient survival. So we want to see on this survival curve whether the expression of another gene might be interacting with T cells to influence overall patient survival. So you can see this is quite similar to the previous care model. And so we test, again, we don't just cherry-pick genes. We test many, many genes. You can see every gene, we can look at their effect. So for example, on this table on the right, you can see PD-L1, which is a well-known this is the ligand the tumor expressed in order to suppress repress the T cell function. It has a positive score, meaning that when PD-L1 is very high, actually the level of T cell even is high, it doesn't really do much. Whereas the bottom gene, which is in blue here, this is actually melanoma antigen gene, which is specifically recognized by the T cells. When it's highly expressed, the T cell is really there to recognize this tumor as an alien and trying to kill the tumor cell. So it has a very negative value in here. And so you can see the value really tells us whether the T cell will work well or not work well. And so we also test every gene in the genome and we give them a score. Even though some are significant, some are not significant, we still estimate a z-score. And when it's not significant, it could be pretty close to 0. And so when a particular patient has a tumor gene expression, we correlate their expression with this overall signal. And here, if it's correlated, it actually tells us the T cells are dysfunctional, because you can see here PD-L1 is a positive score in here. When it's very high, the T cells are there, but it's not doing its job. It's similar to the CBL-B gene. And so if the tumor expression correlates with this genome-wide score, it tells us the T cells are exhausted and it's not working. So immunotherapy may not work. Whereas on the right side, when the gene expression correlation is low, we say, okay, well, maybe the T cell is there and it's not exhausted, it will probably, the patient will benefit from the immunotherapy. And so we do this for many different cohorts that we collected. And so far, what we have seen is that the signature, this is the genome-wide signature, the genome-wide signatures that we derived from endometrial cancers, triple-negative breast cancer, neuroblastoma, AML, and melanoma are actually quite correlated. Also, if we look at those genes that have the very significant this dysfunctional score based on the signal that these positive genes, you can see on the right side, this is a study from MD Anderson recently published Nature paper where the mouse are treated long-term to see the T cell dysfunction. This is long-term treated CMV, so the T cell become gradually dysfunctional 5 days, 14 days, 28 days, 60 days after the LCMV treatment, and the T cells are becoming increasingly dysfunctional or exhausted. And you can see our tide score, our signature score, indeed over time, you can see with time, these signatures become stronger and stronger in the mouse models. And so we also see some other studies. For example, this is mouse studies for T cell exhaustion, Treg, gene activation. A T cell is a screen where T cells were like using shRNA screen, looking at whether T cells would really proliferate and get activated in a mouse, and also mouse treated with CTLA-4. Looking at those differential genes, you can see in those studies, they each have hits versus non-hits, and we ask, can our signature derived from TCGA or these 5 cohorts really predict those hits versus non-hits? And you can see the blue dots and the red dots. Indeed, we can separate the hit. Our score also scored them pretty high for those hit genes. And on the right side, this is also looking at whether we can capture those hits in the screen in those different mouse studies. You can see the signature that we derived from each of these cancer types, they already work pretty well. But if we take their average, which means that we get the most robust signature, in general, we do have better performance. And so this is kind of modeling on the left side before we look at, the T cell is there, but it's not doing its job, it's exhausted and dysfunctional. On the right side, we now want to model T cell exclusion in the cold tumors. And so these are the cases where the T cell cannot infiltrate the tumor. We read some papers, especially Thomas Gajewski's review, where he explained, when the T cell cannot get in, what are the possibilities? One is the MDSC myeloid-derived suppressor cells. One middle one is cancer-associated fibroblast, and then also tumor-associated macrophage. So when in the tumor microenvironment you have high level of these 3, then the T cells cannot infiltrate the tumor. So from the public data, we also extracted the gene expression signatures or the gene expression, genome-wide gene expression level of MDSC, CAF, and TAM, and then look at, for example, in this case is melanoma samples, whether the level of CD8 T cell infiltration is correlated with the MDSC, CAF, or TAM levels. And you can see in general, so each dot here is one sample in a TCGA melanoma tumor. In general, if the tumor has a high level of MDSC or high correlation with the MDSC signature, you will have low CD8 infiltration. Also, when the tumor has high fibroblast, high macrophage, you will have low CD8 T cell infiltration. And then on the very right side is the mean. We average the signature from all 3 together. And then you can see, in general, when the tumor has high correlation with this signature, indeed, it will have lower CD8 infiltration. And the correlation is even more robust here. And so we use this to really model kind of the T cell exclusion when the T cell cannot get into the tumor. And so now let's look at what's the relationship with between T cell dysfunction and T cell exclusion. On the left side, this is all the melanoma samples within TCGA. Each dot is a particular tumor, and the color tells us how much the T cell, cytolytic T cells are activated. So you can see this negative correlation, which means that some tumors have T cell exclusion where the T cells cannot get in. That's on the lower right side. But whereas other tumors, the T cells can get in, and the cytolytic activity is high, but then also you start to see T cell dysfunction when the T cells become too tired. On the right side, we're looking at all the TCGA cancer types. So every cancer type is a different cancer. And so on the right side, this one is lung cancer. This is squamous lung cancer. So in general, lung cancer is a pretty cold tumor. You can see here, in general, the tumor have low T cell infiltration. And on the upper left corner, this is a kidney cancer. Kidney cancer actually is a surprising cancer type that respond well to immunotherapy, partly because its T cell infiltration is very high. It's a hot tumor. And so, so at the end, we put the two models together. We call it the TIDE. It's kind of like the TIDE detergent, which here stands for tumor, immune dysfunction, and exclusion. So we model two things. Dysfunction is modeling when the T cells is in the tumor, but it's too exhausted to kill the cancers. And but then exclusion is the other situation where the tumor is cold and the T cell cannot get into the tumor. So how do we do this? When somebody profile a tumor based on the gene expression, we first decide whether it's CD8 high or CD8 low, or the cytolytic activity is high or low. If it's high, we say, okay, the T cell is infiltrating. Then we use the previous, the dysfunctional score, genome-wide score, to correlate with the tumor gene expression. If it is correlated, we'll say, okay, the tumor T cell is there, but it's too exhausted, so probably it will not respond. But if the T cell is there, but not dysfunctional or exhausted, maybe the patient will benefit. On the left side, when the CD8 T cell is low, we then ask, do we see the exclusion signatures based on the macrophage, fibroblast, and MDSC signatures? And so if the correlation is high, it means that the macrophage or MDSC or fibroblast is actively excluding the T cells from getting into the tumor, so patient probably wouldn't respond. But if it doesn't have the signature, we'll say, well, maybe the T cell haven't got a chance to go in yet, so maybe it will still respond. And so that's how we model them together. And so let's see how it works. We look at the two different published clinical studies. The first is the anti-PD-1 trial. This is from Roger Luo and Tony Riba's lab at UCLA. And so you can see we first separate by the CD8 infiltration level. When the CD8 level or the cytolytic activity level is high, we look at the T cell dysfunction, and you can see as the dysfunction increase, the patient are less likely to respond to immunotherapy. So here, red is responding and blue is not responding. Whereas on the left side, when the tumors are cold, then we look at how much the T cells are not getting into the tumor because of some active repression by fibroblasts or macrophage. And so you can see here, when the immune infiltration is low and the exclusion signature is very, very strong, then the patient usually don't respond to immunotherapy. Whereas when the exclusion signature is not so strong, the patients still have pretty good chance of responding. At the bottom is another trial. This is anti-CTLA-4 treatment that's done in Ellie Van Allen and the Levi Garraway's lab. Again, we see similar trend. When the T cell level is high, we look at dysfunction, and the stronger the dysfunction, the less likely the patient will respond. When the T cell infiltration is low, or the cytolytic activity is low, we look at T cell exclusion, and the stronger the exclusion, the worse the patient survival. We cannot predict this at 100% accuracy. In fact, nobody can predict it at 100% accuracy, but we see from promising results. So overall, you can see for the responders and non-responders for those two trials, indeed, we can actually see a good separation in terms of the overall predictive power. So far, what has been used is mutations, the overall tumor mutation load, and the PD-L1 levels that people have used it in the clinical settings, and you can see the TIDE model can outperform both. We also look at other potential markers that people have discussed, including interferon gamma levels. This is IPS is a computational method, and somatic copy number changes. So in these other methods, in general, we see each is promising to a certain degree, but actually this TIDE model performed the best. Not only we can predict the response, we can see the overall patient survival of the patient over time for anti-PD-1 and anti-CTLA-4, that the patient that have, so either their T cells are too exhausted, or if their T cells can't get in because of those suppressive factors, those patients in general don't do very well in their overall survival, whereas the remaining ones that are more likely to respond to immunotherapy, also they are more likely to survive much longer. And so in summary, we found that the gene expression of treatment-naive tumors from the TCGA already contain information on the patient immune potential in the tumor microenvironment. And so the signatures that we derive from the TCGA without immunotherapy can be used to predict patient response to immunotherapy. And so in addition to actually just deriving a biomarker, from here, we also care about some of the really strong genes, like PD-1, like PD-L1. Some of these genes that can be actually targeted to improve the immunotherapy response. So within our lab and our collaborator, we're trying to validate some of those genes whose inhibition can actually improve the immunotherapy response. So I want to thank members of my lab who participated in the study. The study is led by a very talented postdoc, Peng Jiang. So both the CARE model and the TIDE model was developed by Peng, and he also did the experimental validation for the CARE model. For the TIDE validation, currently Stan Gu is collaborating with Kai Wuki Clinic to do the experimental validation. I also want to thank my other colleagues. John Astor is on the CARE model. Jun Liu is a computational collaborator, our statistics collaborator. Miles Brown is our cancer biologist collaborator. Gordon Freeman is the PD-1, PD-L1 expert. He also gave us a lot of help on in vivo validation. So I also want to thank the funding agency for their support. So yeah, so in general, in my lab, in addition to this computational modeling, we also study using epigenetic profiles and also CRISPR screens to look at cancer in a more holistic approach, because we believe that CRISPR screens and also mutation analysis can give us information about targeted therapy, and these targeted therapy usually just directly try to kill the cancer cells. Immunotherapy is more trying to boost the patient immune system and not directly target the cancer cells, but use the immune system to kill the cancer cells. Whereas with epigenetic therapy, which is also some area we are working on, we feel like it's kind of just tuning the system balance. And so in the long run, we believe only a good combination of the three, these are very complementary approaches, good combination of the three will hopefully help us to reach this cancer moonshot goal. Maybe we don't cure cancer at 2020, but in fairly foreseeable future, we can see most cancer getting cured. So thank you very much for your attention. So that concludes my talk. Any questions? Any questions?
Thank you so much, Dr. Liu, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the bottom left of the presentation window. Type your questions into the box that appear on the screen and click the send button. We'll answer as many questions as we have time for. So let's take a look at what our audience members are asking today. We have a great audience today. Our first question is, in the TIDE model, you first need to decide whether the tumor is cold or hot. So how do you determine the cutoff for this, Dr. Liu?
Really good question here. Yeah, really good question here. How do we decide cold or hot? First of all, I mentioned we use 5 gene markers, the CD8AB, granzyme AB, and porphyrin. So for TCGA, we can look at all 10,000 tumors together, and usually you see a majority of the tumors are fairly cold, but then a subset of the tumors will have a high level, and so there we can use some standard cutoff. However, depending on the specific cohort, for example, in general, as we mentioned, kidney cancers, almost all the tumors are hot, so there we only look at dysfunction, whereas maybe for some other tumors like prostate, most of the tumors are cold, and so there it's true we need a standard. Whereas, for example, other cancer types like melanoma samples, you would see actually some patients are hot, some patients are cold. So in the 2 clinical trials we mentioned, we just take the tumor, we calculate the average level of those 5 genes, and so then if the tumor expression of those 5 genes are above the average, then we say it's hot. Whereas if it's lower, we call it a cold tumor. But indeed, I think, so for melanoma, this might be an easier question, but for some other tumors, exactly where the cut line could still be pretty tricky.
Thank you, Dr. Liu. We have time for one more question, and we have a wonderful question from one of our audience members. In your care model, you use differentially expressed genes between sensitive resistant cells as the gold standards. Is this a good choice? And also, you mentioned that the care performs better than the CRISPR screens, which some people might consider as a better gold standard. Can you explain this a little more? Thank you.
Another very, very good question here. About the gold standard, we do get asked by reviewers, why do you use a cell line, sensitive, resistant line, and look at the differential genes? Unfortunately, right now in the cancer field, nobody have real gold standard. And so we would say using cell lines chronically treated with the drug to derive a resistant line and looking at differential gene expression, it's a kind of a proxy of a gold standard. I wouldn't say it's perfect. It may be like a silver standard. I would say it's not the best, but we don't really have better standard. Some might argue that CRISPR screen should really be much more important. You treat the cell. So first of all, you use CRISPR to knock out different genes, and you treat the cell with the drug, compare with the cell without the drug, and you look at sensitivity of the cell to the drug when different genes are knocked out or knocked down. Somehow in our analysis, we found it to work, the care model to work even better than CRISPR, which is a little surprising because right now our TIDE model was generated on compound screens over hundreds of cell lines. For the same drug, we look at 100 cells, how the cell will perform based on the expression of those or the mutation status of the cell. Whereas CRISPR screen right now, it's still very limited. I know people are generating a lot of CRISPR screens right now. The initial, most of the published screens so far were just on baseline cell lines, not treated with any drug. And so we are only looking at very limited the CRISPR screens on a single cell line and look at the CRISPR result. We believe if you have hundreds of cell lines treated with the drug and then followed like a CRISPR screen with that drug, we should be able to derive a good model, a much better, I would say, potential biomarker for the therapy response. And so CRISPR only doesn't work that well when you profile on a single cell line. With more cell lines, it should work better.
Dr. Liu, I want to thank you again for your wonderful presentation and truly your important research. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind the audience members that today's webcast will be available for on-demand viewing through January 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.